Hi, I'm Matt Gazzara from VOILA Lab, and I'll be leading this part of the talk. But before I start, has everyone been able to log on to the virtual machine who wants to log on? Good? We're all good? All right, so. Right. Okay, so Yosef gave you an overview of all the things that can be done with MAJIQ. Now we're going to kind of get a little more hands-on and talk, practically speaking, how do you use it in different analysis scenarios to get the kind of results that Yosef just described to you all. And along the way, feel free to ask any questions. We'll do some hands-on steps, so we'll take little breaks for executing those and troubleshooting all of that. Yeah. So what kind of analysis scenarios might we want to accomplish? Yosef mentioned before, common things you want to do is analyze psi. What are the relative inclusion levels of different isoforms within an experimental condition, or delta psi if you want to compare between two experimental conditions? How large is your data set? How many samples do you have in groups? How many groups do you have is something that we'll consider. Do you have replicates or not? We can handle analysis with and without replicates. Once you start defining replicates, there's some things you should keep in mind. We'll talk about that. Do you suspect that you have outliers in your data, or do you not have like true biological replicates? Is one of your samples mislabeled? Are you dealing with more heterogeneous data? We'll touch on that. Do you have paired samples? Maybe you have patient data from two different tissues and you want to do analysis comparing those tissues within a patient and then generalize that across several patients in which you have paired samples. That's something we'll talk about. Is your data more heterogeneous? Do you have large patient populations? How can you use MAJIQ to get splicing information over this kind of data set? We'll talk about that as well. Related to the heterogeneity, if you define experimental groups within patient populations, you might want to look at splicing variations within a group versus between groups. This would allow you to capture some of that heterogeneity that you might expect in human patients. And also, MAJIQ, usually we assume you want to analyze the entire genome, but you can also make things really quick and sped up if you only have a relatively small gene set that you're interested in. So that can make things very fast. We'll talk about how you can accomplish that. And we'll mention throughout kind of high versus low coverage, how might that affect different steps of the analysis. And then finally, we'll say, once you get these results, how can you interpret them with VOILA and go to like experimentally validate or look further into splicing events that you find to be differentially spliced? And what does that mean biologically or physiologically in your condition of interest? So the basic MAJIQ pipeline looks like this, and Yosef mentioned this before. So in this workshop, we're going to assume that you've already mapped your data. The data on the cluster is BAM files that are sorted and indexed already, so you can choose your favorite mapper to map. We prefer star for the speed and accuracy. So given your annotation database, a GFF3 file, and your RNA-Seq reads, the first thing you do is run the MAJIQ builder. This defines your LSVs, who's connected to who, which junctions are reliably detected. That's done in this step. Next, you run the MAJIQ quantifier. There are two different ways to run that, either psi if you're only looking in one experimental condition or group, or delta psi if you're comparing things. And finally, you use VOILA to visualize the results that the MAJIQ quantifier put together for you. And just like MAJIQ, you can visualize things for psi within a single condition or delta psi between two conditions that's illustrated here. So that's just like the overview of what happens, but let's kind of walk through what kind of files you'd actually see and what the commands might look like, just to give you, a broad overview of where we're going before we get into like the details. So in our example that we'll be working with today, we're dealing with mouse tissues. We have mouse cerebellum and mouse adrenal gland. These are BAM files, so they're binary SAM files that have been sorted and indexed. So you have the BAM file that contains the reads, and an index file that will tell MAJIQ which points to access that file for the specific reads. So this is one replicate in cerebellum, one replicate in adrenal gland. Let's say we have replicates for these. So we've got cerebellum from 3 different mice and adrenal glands from 3 different mice. That's what our data looks like. So the first thing we'll do, like I mentioned, is run the MAJIQ build that detects the LSVs that are in this data. So the output of this, once you feed it your BAM files and the annotation database, are these files here. You have a single splice graph file that contains all the coordinate information, where the junctions in the genome, where the exons, who's connected to who, and that's used for visualizing later. And you have all of these MAJIQ HDF5 files that contain the bulk of the information. That's the actual quantification. So now at this point, our LSVs have been defined. Next, we want to quantify those LSVs. In this toy example, we're comparing between 2 experimental groups. So we'll run MAJIQ delta psi. And all you need to give the delta psi quantifier is your MAJIQ.hdf5 files that come from group 1. We're calling group 1 cerebellum here. And your MAJIQ.hdf5 files that come from adrenal gland, group 2. And you tell it what the names of those 2 groups are, and you'll get going. It'll run, right? So group 1, group 2, and the output of this step is the important output of this step is a single voila file that contains all the quantification for these 2 groups. So now our LSVs are quantified. The final step to make this interpretable, unless you want to read this pickle file that has a very particular data structure, is to run it through voila. So voila takes the voila file that the MAJIQ quantifier produced and the splice graph that we made back at the builder step, and it produces the output. So we have an index file that's kind of your gateway into navigating the results. You have a number of summary files. There are 3 here, but you could have hundreds or even thousands if you have many, many LSVs. This contains the actual per gene results, 10 genes per page, and a tab-separated file that's useful for parsing and doing kind of genome-wide analysis with any Python or similar scripts you might write. So now our LSVs have been visualized. So this is kind of the basic workflow, just to give you an idea before we get into the nitty-gritty and the steps and the parameters that you can adjust at each of these stages to change your results. Yes?
We're using a GFF3 file.
Yes.
Input for the LSVs.
Mm-hmm.
Are you doing de novo as part of this workflow?
Yes, so by default, de novo detection will be done, and I'll go over how that's done, but you have the option to turn that off, and we've got a slide on that. Obviously, that'll speed things up,
So
but
if you're including GFF3, though, you're using
yes.
those as sort of the starting point and then building.
Exactly, yeah, exactly. And really, technically, MAJIQ can work if you just give it the boundaries of genes. That's what one key piece of information we use from the GFF3 file. Where does the gene start and end? And then we look for any splice junctions within that boundary and start building on top of that. So technically, if you only gave us the gene boundary, we would in theory be able to recreate the transcript without the rest of the annotation. But you're exactly right. We start with the annotation database and all the junctions and exons they say as a starting point. We see what we think is there reliably, and we supplement it with de novo things that exist reliably within your data to expand things. Right. So we'll go over each of these main 3 steps and mention the different parameters there. So the first step, like I mentioned, is the builder that defines your LSVs. This is the basic command line that I've alluded to. You're doing MAJIQ build. You give it your annotation database, a GFF3. We'll go over what that format means. You give it a configuration file that just kind of does some bookkeeping on where your files and genome is. And you tell it what your output is, and that's all you need to do to get going. There's some options that we'll cover as well. So these are some of the considerations we'll cover for the builder stage that you might want to tweak depending on what your data is like. So the first thing I want to just mention is the annotation database. It's a GFF3. On the website, we've got GFF3s that we've used for human HG19 and mouse MM10 genome builds. But the links here also describe what the general format is. So if you've got another organism, you can use that. It's a fairly common file type that you can download from most databases like Ensemble, Flybase, Wormbase, and you can get going. But I just want to point out like some of the key features and some of the key things that MAJIQ expects to see when it gets the GFF3 file that'll influence what you actually analyze downstream. So the third column here is like the feature column that tells you what is this line describing. So MAJIQ, I've bolded here the things that MAJIQ will pick up on and some things that MAJIQ doesn't really care about. So for every gene in the GFF3 file, there should be a gene line that's defined here. That gene should have a unique ID or a unique name, and that defines the boundaries of where this gene, RBMX, occurs in the genome. Next, associated with that gene, you have mRNAs or transcripts that say this is a particular version of this gene, this transcript that we see. It has a parent attribute here that tells us it is related to this gene ID here. So there's a hierarchical structure here, and then under mRNA or transcripts, you have exons. And these exons have a parent attribute that's associated to the transcript from which they came. Yes?
So a gene is a maximum boundary of all the mRNAs.
Yes. Yep.
So what if it has two mRNAs that transcribe in different directions?
In different directions. So if you wanted to pick that up, so a few things. If you have a strand-specific library preparation and there's some overlap, you should be able to differentiate which came from the sense and the antisense strand. But MAJIQ doesn't have problems really with overlapping transcripts, given that you've done strand-specific analysis. You should have kind of another gene entry for the antisense strand in theory that defines it slightly differently, but yeah. So I think this is a bit better illustrated with this kind of tree-like structure. So MAJIQ looks for a gene that defines the outer boundaries, one or more mRNAs that are associated with that gene, and mRNAs are made up of exons. And later in the file, we've got another new gene, PTB protein 1, that has an mRNA and a transcript associated and a bunch of exons. So this is the kind of like the minimum requirements of what should be in your GFF3 file. Practically speaking, this is a step that a lot of people tend to have problems with, so we wanted to mention it. But yeah, just to recap, genes, the boundaries, mRNA or transcripts defines the transcripts associated with that gene. Again, they need the parent attribute here to associate it with the gene, and the exons should be associated with a parent transcript. So this is really important if you kind of want to restrict your analysis to a very small, question, yeah.
Does the GFF3 file have to be sorted at all, or can it just be?
It doesn't need to be. I think it works faster if it's sorted, right? The parsing of the first step in the builder is it goes through this file and parses it. The parsing will be faster if it's sorted, but we don't necessarily assume that it's sorted, right? As long as the parent attributes are correct, we'll find it and link everything together in the right way.
the only requirement is that the bag attributes are defined before anything is below it. For example, when you define the exon, you need to be defined an array before that exon and the gene before that.
Oh, okay, yeah. Yes.
So you're throwing away UTR and coding sequence information?
Yes, exactly. Yeah.
Splicing would be happening in UTR.
Well, so the thing is, most annotations are somewhat redundant. So UTR, and these coding sequences here are already put in here as in the exon coordinates. It's just kind of like another feature on top of that. So like this exon here, we're saying that's an exon from this position to this position. This UTR attribute just tells us, oh, this exon is annotated as a 3' or a 5' UTR, right? So it's redundant information in a way. Same thing for the coding sequence. So for every exon, you'll have definitions of what the UTR, or if it's coding sequence, where the stop and start codon is. This is all information that may be in your GFF3 file that we kind of gloss over and don't pay attention to. So if you want to analyze things that are kind of like that, you can tweak the GFF3 file to call it like an exon, and that's what MAJIQ takes note of. Although, like I said, it should be redundant. So if you want to analyze just a small gene set of interest to speed things up, just delete everything in the GFF3 file that's not in your gene set of interest, and it'll make things go very quick. Like I mentioned, just make sure you meet MAJIQ's assumptions that you have genes that are associated with mRNA, or that have mRNAs or transcripts associated with them, and those mRNA or transcripts have exons associated with it. So if your GFF3 annotation has in the feature column things like link RNA or tRNA or other feature types, we're going to not analyze those in the builder. All you have to do is kind of change it to transcript, and we'll pick it up and we'll look for splicing variations there. Yes?
What about circular RNAs?
So we don't focus on circular RNAs right
It was
now.
in the list of things we said we're not doing.
But what I mean is, if, say, I have a circular RNA, and algorithm of VOILA, or analyze as
We've never tried it. I wouldn't expect it to work because most of them are like back splicing reactions, right? So you have a read that starts downstream and then splices to upstream. I don't think we formally tested it. I don't know if Jordy knows that that'll definitely fail.
I don't know, because that's a good point, because we expect that the junctions goes always forward in sequence. And we expect that the junctions are always splicing forward so the start of the junction and the end of the junction is going to be always in a forward direction. If we splice someplace before in the transcripts, I don't know if it worked. I'm not sure. Something it's worth it to try. But I cannot tell you what would happen right now.
it's not definitely
It's not going to crash, but it's no. I don't know how good can you trust that result. It's something worth it to try.
Yeah, we honestly haven't tried it yet. I wouldn't expect it to work. If it doesn't crash, I don't know that our quantification or what we detect would be that great, but it's something we should look into, yeah. Right. So that's the GFF3 file. Something to keep in mind that especially, and we'll do this for the toy execution we've got here, MAJIQ empirically determines a number of normalization factors based on what you see in the transcriptome. So one of those normalization factors is to normalize for local GC content. And to empirically determine that, it looks for what local GC content is for all junctions in the genome and applies a correction factor. So if we're going to analyze a small gene set of like 1 to 20 or 100 genes, we want to invoke the no GC option here, so it will not apply that GC content correction. If you do it on such a small gene set, the normalization factors there can really kind of throw off the quantification. So this is something you definitely want to invoke if you're doing a tailored gene set. So that this is what the execution would look like. You just add the no GC option at the end. Very simple. So the other key part of what you need to feed the builder is the configuration file. We've got this on the cluster, but it has this general format. You've got an info section that has like some bookkeeping information of what your RNA-Seq data is like. One thing we need is how long are your reads, the length of them, to initialize the arrays where we count where the reads actually start. Paired end, singled end, it doesn't matter. You just, yes. Yes. If you have variable length reads, you just, in this part, you put the longest read that you have in your data set you're going to run through the builder. So if we had mixed like 101, 76, 36, I wouldn't recommend that. You would just put 101 because that's the highest one. And if you have 101 paired end reads, you just put the length of one of the mates, not both. So star will say your read length in this case would be 202 because it considers both mate pairs. But for the builder, you just need the length of one of those mates. Yes?
So if we had like 3 lanes of sequencing, 2 are 50 base pairs, and 1 is 125 paired end, just put 125 there?
Yes, yep, exactly. So that's read length, and everyone brought up great issues. Those are all the things I wanted to cover there. SAM directory is the directory where your sorted and indexed BAM files are. They should all be in the same directory currently how it's implemented. So if you've got data kind of all over the place, we don't support that. Just throw it in one folder and run it. Genome annotation is just important for some of the output files. We link to UCSC and things. This isn't that important, but it'll give you connectivity to things like UCSC Genome Browser later down the road. And finally, or second to last, genome path. This is the path to the genome FASTA files, the sequence files. This is where we get the nucleotides for local GC content correction. So for our toy example here, that file is just going to be empty because we're not doing GC content normalization. But you do want to have a version of the genome downloaded somewhere so we could apply those filters looking for local GC content. Finally, if you're dealing with strand-specific data, you just have this type equals strand-specific here. That'll tell us to only look for reads on one strand or the other, not both. But for today, we're not dealing with strand-specific data, so that option is just left out of the configuration file. That's the info section. The experiment section, here we have 2 experimental groups. Experimental groups are defined here at the beginning of the line with an equal sign, and then we have a comma-separated list of replicates in this experimental group. So we have a group we're calling ADR for adrenal, and that group is composed of adrenal 1 replicate, 2 replicate, 3 replicate, and so on. These names here should match the BAM files that are located here. So MAJIQ's going to look for adr1.bam and adr1.bam.bai, that's the index file. And the same for cerebellum. So one thing to really keep in mind here is the builder, we expect you to kind of like run it one and done. You run one big execution that contains all the experiments you might want to analyze, and then downstream for delta psi and psi, that's where you run like your specific pairs. So here we've got 2 tissue groups, but maybe we have 12 different tissues that we want to analyze against each other. We would have 12 tissue groups here, so MAJIQ looks over all of the data you'll ever be interested in analyzing, defines the LSVs in a common way over all of that data, and then later on in delta psi or psi, you say, oh, well, I only want to look at these 2 specific tissues or these other 2 specific tissues. So that's a key point. You want to build everything together.
So conceptually, coming back to the previous, the reason to do this separated like this is you have one builder that will identify all the variations across all your samples, so they all speak the same language, right? Because there's a lot of de novo stuff that are not necessarily annotated, and you want to make sure all the LSVs that you detect in all the samples are all, talking about the same LSVs. So this is why you do the builder, you do it once, and you do it with all your samples. And then, once you've done it once, you can do many different psi and delta psi quantifications for various groups or various analysis that you're interested in. So that's sort of the logic behind those constraints. Yeah?
Okay, so now we've talked about what the transcriptome database and the configuration file should look like. Another thing to consider are the reliable filters that Yosef's mentioned before. These are filters that apply when we initially detect LSVs to kind of see what exists in the data that you're feeding the builder. So an LSV to be considered reliable needs a junction that has at least 2 positions that are covered. So that means the reads that span the junction should start at 2 separate genomic positions. That gives us confidence that it's not something like a PCR stack or something. We're hitting the junction with multiple reads in multiple positions. The minimum number of reads we should see at a junction is 3. So these 2 parameters must be, or these 2 thresholds must be met for an LSV to be considered reliable. So these are parameters that you could tweak. With some testing, these were the ones we settled on. But for your data that might be higher or lower coverage, maybe you want to alter these and play around with them a little bit. For de novo junctions, we have a little bit different requirements. We look across all the replicates in an experimental group, and we want to find an average of at least 2 reads across that de novo junction that's not in the annotation database for it to be considered a possible de novo junction. You could alter that float value by using min de novo and altering the 2 there to something else, a higher number if you want to be more confident, or a lower number if you want to be more permissive. So now these filters, you look at each sample individually in an experimental group, and these filters must be passed, these thresholds must be met in at least 51% of the replicates in an experimental group to be considered reliable. So that means that your definition of experimental groups can influence what you ultimately detect in the builder stage. So, with that in mind, you might have one replicate that was relatively low coverage compared to your others, but you don't want to just merge all your files together and lose information from your different replicates. Or you're dealing with things that are not true replicates where, heterogeneity and rare junctions might be expected. It might happen in 1 patient out of 10, but you've got 10 patients that you've put in an experimental group, so you'll not detect that as a reliable LSV. So our solution is you can alter your configuration file when you define your experimental groups. The one I mentioned was this, where we define a group with replicates, but you could also make each of these samples its own experimental group, so you'll have more detection power, you'll detect a lot more LSVs. And just using our toy set of 20 genes, these are the number of LSVs that we find when you define things as groups versus when you keep each sample as its own experimental group. So similar to that, we have a parameter called min experiments, where you can just tell MAJIQ that you don't need 2 out of 3 to find an LSV reliable. We could change that to 1. So depending on what your samples are like, you can play with these thresholds or the configuration file to maximize your detection, or on the other hand, be more conservative if you want to do either. Yes, you can do either. They're very similar. The behavior is very, very similar.
Okay, so notice the original definition of the 51% in a group is the reason for this is, we tend to develop this to our own needs, right? We're very centric in that sense, and we did this because when we did this initial analysis, we wanted to be very conservative when we did this genome-wide view for the eLife paper about what we report. We didn't want to report junk and say, oh, there's complexity, and then somebody goes and say, hey, those are the things are artifacts. So we set a very conservative threshold. If you look at it, if you think, we have 6 or 8 replicates, we want 51%, at least half of them, to be reliably detected in order to say, oh, this is real, I'm going to include it in my genome-wide view. But maybe your needs are very different. Maybe your data is more heterogeneous. Maybe you want to be more lenient in your definition. So this is why really keep that in mind. The default thresholds are not magic. Okay, it was derived based on what we were trying to achieve, and we were trying to be conservative. So I want to, so when you do your analysis, figure out what works best for you. Yeah.
Can we have overlapping? Can I have all from left and right all in the
Yeah, you can. But you should understand that the main difference between, as Matthew said, the difference between putting in one group or putting in separate groups is that you're going to find more LSVs in separate groups, but the LSVs you'll find are less confident ones. So everything you do in replicas is going to have a higher confidence on their detection. You're going to find more LSVs, but the extra ones you're finding, separating them, are probably less confident.
What I mean is, when it's reporting, if I say I scramble my 6 things in some random order, define 2 other groups, right? So when they're reporting the LSVs, they're going to report not just in terms of sample, but also which sample belongs to which group.
You're talking about delta psi when you scramble your
No,
group?
he talks about the builder.
So I define another group, like the tri group equals to SIR1, ADR2, SIR3. When it's reporting, it will reporting the LSV in that group for.
Yeah, you're right. If you put adrenal 1, 2, 3 in a group, and then you put it as well, adrenal 1, that's gonna, you should not do that, because at the end, the output files of the builder is based on sample.
So it's not going to
It's,
distinguish.
it's gonna probably gonna overlap each other.
Oh, okay.
But it's something we want to change, since we want to make a different approach and create output from the builder based on groups that you define.
So if I want to have overlapping group, I should just run another
Yeah, another builder instance.
one. Okay.
Yeah. Yeah,
Right
those,
now,
yeah, good questions. So Yosef mentioned this before, but there is also a non-de novo option if you don't want to detect things de novo in the RNA-Seq data. You just invoke the option -- non-de novo. This speeds up the execution time greatly, but you'll obviously detect fewer LSVs if you're not looking for de novo junctions. So this is just a comparison with de novo in our set of 20 genes, a very small set, versus non-de novo, you find many fewer LSVs. Importantly, when you're looking at kind of patient data or knockout conditions that probably haven't been queried when building these annotation databases, you miss some really biologically interesting and relevant de novo junctions that occur only in certain patients. This example is from a paper we published with Andre's group that describes how skipping of this exon 2 allows cancer cells to escape CAR T cell therapy. But kind of practically speaking, knowing that non-de novo speeds up execution a lot, one scenario we've done before is kind of do a discovery phase where you have some high confidence, high coverage experiments where you kind of see which junctions, which LSVs are available, use that to build your own custom annotation database, and then run non-de novo on a large set. If you have many samples where you want to do hundreds of pairwise delta psi comparisons, that would take a very long time with non-de novo, or sorry, when you're looking for de novo. So that's a way to kind of get around and maximize the speed of the execution. So that's something you might want to consider a use for non-de novo. So finally in the builder, I just want to mention Intron Retention. There's a threshold that's very sensitive to how deep your coverage is. So before I explain what the threshold is and how to use it, this is just a quick summary of how we detect intron retention. So here we have 2 exons and an intron. We divide the intron into 10 equal-sized bins, and we look for average coverage over each of these bins. That average coverage in each bin should be greater than or equal to your set threshold, and then we consider this intron as potentially retained in your data. So with higher coverage, you're going to expect to see higher average read count over these bins. So by default, we look for an average of 1.5 reads per position. And from our paper, this is just altering that threshold, being less conservative to detect introns. You obviously detect many more. So if you have a very high coverage experiment, this was done with about 80 million reads per sample. If you're going above that, you might want to alter the intron retention coverage threshold to kind of tune your detection for introns. Yes?
Would you suggest changing that threshold if, for example, in Arabidopsis, intron retention is the main type of alternative splicing? So you want to detect more of that? Would you recommend to change that or just keep it?
We haven't tried Arabidopsis data.
Yeah, you should tell us.
Yeah.
You're Brian Gregory's lab. No, I know Sean used it, and we analyzed some data, and it seemed to have worked very nicely for him. He was very happy with the results. But that said, we haven't done that much of plant biology, and so we don't know. We think this is, so I would just point out that this is currently what's implemented. We don't think this is ideal. We have ideas to improve it, and hopefully some of those things will be adjusted automatically and a better algorithm will be. But right now, I think, it really depends on, do you really want to, so for example, if you're looking for changes in the protein, you really want to capture those, maybe these are like splicing noise or irrelevant variants that are, degraded, and you don't want them to clutter your visualization in VOILA, then you increase it, right? So I don't know for your specific applications, so you'll have to,
Yeah, and another thing to keep in mind with how we detect intron retention, some people have assumed that this should kind of be a proxy for maybe detecting novel polyadenylation sites or 3' UTRs, right? So an intron might be retained and there might be sufficient polyadenylation signals within the intron to have this transcript cleaved and polyadenylated at this position. But we will not detect that as an intron retention because you'll have a drop off of reads at the point of cleavage and polyadenylation. So it's not designed to detect those, and that's why it won't detect those. So keep that in mind if that's really what you're interested in doing. Our tool's probably not best for that. Okay, so those are, as we see them, some of the main things you should keep in mind when running your builder execution. I guess Jordy would, well, we could, actually, I have it up here on the slide. We can try our first hands-on thing for you guys to actually run who's connected to the cluster, to actually run something and see if you could get it to work. So the first thing we're going to try is what I initially described to you. We're going to compare two groups, each of which have 3 replicates in them. So this is what the settings files looks like. It's already on the cluster, and this is what the command line will look like that you can run. So just to go over the components, so we're going to call MAJIQ as an executable. We're going to tell MAJIQ we're building, so detecting, defining our LSVs. The first thing, we're pointing the builder to the path to the annotation database. This database contains 20 genes of interest, so we're restricting analysis to those 20 genes. This is the path to that file. We're telling the builder where the configuration file is. That's also located on the cluster here. We're telling the builder where the output should go. We're suggesting you call it like build groups. It'll make a directory in your home directory and put the output there. Nthreads tells the builder how many threads to use. Because we're only using 20 genes, only 1 thread's necessary. But if you're doing this on some high-performance desktop or a core, you can up those threads when you're doing genome-wide analysis. And finally, because we're only analyzing 20 genes in this database, we want to invoke the no GC option so the GC content normalization doesn't throw things off. So I guess try and do it.
If you try to run it, the execution of these 20 genes is going to take around a couple of minutes when I did it.
Will it be faster if I use higher number of threads?
No, you want to be nice to your other
Yeah, you
people,
cannot
right?
use
Because
one of our threads as well because we only work
yeah, we're all sharing that virtual cluster that Anand, created for us. Which is one of the reasons we limited it to, 30 participants.
And the thread division is based, I think, on how many genes are in your database, right? That's how you allocate.
There is 2 types of division during MAJIQ. The build, there are 2 internal steps. One is analyzing the BAM files, and it divides the threads based on the number of BAM files. The second division is after it does that, it divides by the it divides by the number of genes there are. I have a lot more. So it's fine.
But if I have my own machine, if I run it, this, so if I have my own machine, if I run it, I should use higher numbers, right?
Totally, yeah.
And it will not distinguish between thread within nodes and the different nodes.
It is, right now it's thought to be run in a single node.
Huh?
In a single node. So you can run up to 16, 32 threads in the same node.
Yeah, but if I have threads number bigger than the supported I have core,
Yeah, you can do it.
then it will find another core.
Yeah.
Right?
Yeah, that's totally fast. The only thing is not done, is not thought that, and is a feature one we are working on, is to be able to work in a distributed environment. So right now it's going to work in the same node with as many core that you specify.
Okay.
Yes. Yep, you should be able to.
It's like a couple, 3 months.
Okay, yeah.